So I'm going to address some of the most common muddiest points I received from the class. So one of the questions that came up a few times is why gluconeogenesis and when do we do it? So for gluconeogenesis, you're making glucose from non-carbohydrate precursors. You're basically doing it when you haven't eaten. Because eating is a simple way that you provide glucose to your body and a simple way that your blood glucose levels are maintained. Now, if you don't eat, you're not going to keel over because your body will make sure that your blood glucose levels stay constant and the glucose is made. So you can see from this picture over here that glucose 6-phosphate is a precursor to all sorts of things, specifically glucose like we talked about in class, but it's also a precursor to building your glycogen stores or making sugar modified proteins, two sugars connected, other single sugars not counting glucose, and other, like with starch, polymers of glucose, and then also sucrose. So basically for us, I think we're going to focus on the fact that gluconeogenesis creates glucose when you haven't eaten. And this is important because I mentioned in class in the very beginning of the lecture that there are some parts of your body that use glucose either exclusively or primarily as their fuel source. Another question that I got pretty frequently is, what do I mean by reciprocal regulation of glycolysis and gluconeogenesis? So I said that these are reciprocally regulated, meaning that when one pathway is happening, the other one isn't happening to the same degree. So when one pathway is activated, the other pathway is inhibited. That's another way to say it. So one thing to keep in mind is that gluconeogenesis only happens in a couple of places in our body. Primarily, we're talking about the liver doing gluconeogenesis. Most places in your body can't do gluconeogenesis. So when your liver is tasked with the job of providing glucose to the blood, and your glycogen stores are very low, your liver will start doing gluconeogenesis to get that glucose to the blood. So if that's the priority, then you don't want the liver using that glucose a lot for its own glycolysis. So glycolysis in that moment will be inhibited to some degree until you're done making the glucose for export, and then the liver can go back to doing glycolysis. The rest of your body doesn't do gluconeogenesis. So this reciprocal regulation is really only relevant when we're talking about a place that does both pathways potentially. Now, people asked for an example of this reciprocal regulation. So we'll talk about regulation in a lot more detail later when we get to the lecture called metabolic regulation. But for now, I can give you an example. So going from fructose 6-phosphate to fructose 1,6-bisphosphate is a step that happens differently whether you're talking glycolysis or you're talking gluconeogenesis, because these steps are not reversible. So in the direction of glycolysis, fructose 6-phosphate is catalyzed to fructose 1,6-bisphosphate by phosphofructokinase. To go in the opposite direction, which happens in gluconeogenesis, a different enzyme is used, fructose 1,6-bisphosphatase, to go from fructose 1,6-bisphosphate to fructose 6-phosphate. So an example of the reciprocal regulation is when you've got plenty of ATP. ATP will act to negatively inhibit or negatively regulate phosphofructokinase 1. Why have that enzyme work in glycolysis when you don't need ATP? That's one of the big picture goals of doing glycolysis, is that you're producing ATP. And if there's plenty there, ATP can act as a molecule that signals phosphofructokinase 1 to just stop working as hard. Similarly, well, I should also say that if that one's inhibited, then in comparison, the fructose 1,6-bisphosphatase can work. So you inhibit one and don't inhibit the other, you're going to favor the pathway that's not inhibited. You definitely wouldn't regulate the flux between these two pathways by affecting an enzyme that catalyzes a reversible step, because if you were to inhibit that enzyme, you would inhibit it for both the pathways, and then it wouldn't really help to promote one pathway and not the other. We'll talk about this in more detail later for sure. But another example I want to give you is the molecule AMP. AMP is what you get when ATP has lost 2 of its phosphoryl groups. And it's a sign that you are low on ATP because it's an ATP breakdown product. So AMP actually has the ability to act on both of these enzymes. AMP will bind to phosphofructokinase and activate it. It can also bind to fructose 1,6-bisphosphatase and inhibit it. When you're low on ATP, don't be spending ATP to make glucose. You can't afford it. Let glycolysis take center stage to make a bunch of ATP, and that is the important thing for that moment. The key is it's moment to moment. So at any point, ATP levels rise and then glycolysis gets inhibited again. So this is called flux. The next most common question was about bypass reaction 1. We looked at this figure in class, but let's add some more to create context. So the PEP is going to be converted in the next step to 2-phosphoglycerate, which then goes to 3-phosphoglycerate. I'm only drawing this in the direction of gluconeogenesis for simplicity's sake. And then we go to 1,3-bisphosphoglycerate. And then we go to glyceraldehyde 3-phosphate. And we go to fructose 1,6-bisphosphate, fructose 6-phosphate, glucose 6-phosphate, and then glucose. And I want to point out that to get from 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate, you need some NADH. So NADH is needed in this step. In the opposite direction that happens in glycolysis, you make an NADH. But in gluconeogenesis, you need one at this step. So that can help us understand why we evolved to have a path that looked like this. You start with pyruvate, which gets moved into the mitochondria. And is converted to oxaloacetate. Now, for things to get out of the mitochondria, they have to pass the inner membrane. And to pass the inner membrane, you need to have a protein transporter that allows you to pass through that lipid bilayer. In this picture, they're also showing you the outer membrane of the mitochondria, which has large pores and lets things pass easily. So it's really the inner membrane that's the selective barrier here. So there is no oxaloacetate transporter protein, so that molecule can't get out. That molecule is the precursor to PEP, though. So somebody asked, why would we bother making oxaloacetate? It's the step that you make before you get to become PEP. Remember that metabolism is just lots of incremental steps toward what you want to get to. And so you have to make oxaloacetate. But we evolved to have a path where oxaloacetate is made in the mitochondria, but it can't get out, so that it gets converted by malate, it gets converted to malate by malate dehydrogenase. And this requires the reducing power of NADH. And effectively, you can think about that reducing power being transferred to oxaloacetate, and now malate has it, so that when malate leaves the mitochondria and it's in the cytosol, it can cause the reduction of NAD back to NADH so that oxaloacetate is formed. That seems like a bunch of unnecessary steps, but we evolved to do it this way because it provides NADH to the cytosol, and if you're doing this first few steps of gluconeogenesis, you're going to do this later step over here that requires NADH. So you're setting yourself up for future success by doing something in the beginning of the pathway that helps a later step to happen. I also mentioned that alanine is a precursor to pyruvate. We'll talk about this reaction in more detail later. All we need to know here is that this amino acid can be converted to pyruvate so that there's pyruvate to enter into gluconeogenesis. In case you're wondering about the significance of alanine, we'll talk about it more later, but when proteins get broken down in your muscle, because you're starting to use muscle for fuel, which isn't ideal, but it helps you survive, all of those amino acids are converted to alanine and travel in the blood to the liver as alanine, and that way can feed into gluconeogenesis. So we can write that here as from skeletal muscle. But we'll get into that path a little bit later. So bypass step 1, which involves a couple of reactions, can also have a slightly different path. Here we see the path that happens when your body is producing lactate. You're normally not doing very much of this pathway because you're not producing much lactate normally. You only produce lactate when you're doing anaerobic respiration in your skeletal muscles. The lactate will travel from the muscle to the liver, and that lactate will get converted to pyruvate so that gluconeogenesis can happen. And in this path, you don't have malate being involved because this step of lactate to pyruvate conversion creates NADH in the cytosol. Now, this is an error in their figure. Let's fix that. So remember how I said that you need NADH in the cytosol for a later step? Well, when you're using lactate, you make that NADH straight away from that first step. So this path does not include a step where malate gets formed and then gets turned back to oxaloacetate. The pyruvate is moved into the mitochondria, converted to oxaloacetate, and then directly converted to PEP, and PEP leaves. So there's some interesting things to note. There's a mitochondrial malate dehydrogenase and a cytosolic malate dehydrogenase. So that reaction happens in both the inside and outside of the mitochondria. You also have a carboxykinase, a PEP carboxykinase that's in the mitochondria. And that same reaction happens in the cytosol with this PEP carboxykinase. You might be wondering, what would happen, or does this happen? This oxaloacetate turns into PEP through that PEP carboxykinase rather than getting turned into malate. That would be a problem because that's not going to add NADH to the cytosol like we just talked about. So could that happen? Well, it looks like it could happen in this picture, but in reality, there are mechanisms in place so that the oxaloacetate does get converted to malate if it originated from pyruvate. So you're usually doing this path where you're originating from pyruvate or from an alanine that turns into pyruvate. You're not doing the lactate path very often because you don't produce that much lactate, and you don't produce lactate often, only from anaerobic respiration. And when that happens, then the path on the right will happen. One thing to notice, now that we have both these paths right next to each other, is that pyruvate carboxylase only exists in the mitochondrial matrix. So that step from pyruvate to oxaloacetate is only occurring in the mitochondria. And lastly, I had several questions about the comment that fatty acids are not glucogenic. They can't produce glucose, and neither can leucine or lysine. So the point here is that these three things cannot contribute to a net production of glucose. Fatty acids get converted or get broken down to acetyl-CoA. Leucine and lysine also get broken down to acetyl-CoA or a related molecule called aceto-acetyl-CoA. We'll talk about these molecules a lot more later in the course. So you might think, okay, acetyl-CoA, that enters into the citric acid cycle. And we know that a lot of the citric acid cycle intermediates like oxaloacetate are precursors for glucose. So why isn't acetyl-CoA a precursor of glucose? Why do we say it cannot feed into gluconeogenesis? And that's because it does not result in a net production of glucose. The two carbons from acetyl-CoA are lost as CO2 during the Krebs cycle. That's the most direct explanation that I've heard about it. If you look online, you see a lot of different types of reasonings as to why fatty acids don't feed into gluconeogenesis. Hopefully this has cleared up some of the major muddy points. Please let me know if I could answer more questions by posting on Piazza.